Hello everyone, I'm here with Dr. Lien from Yangming University. So, Dr. Lien, can you tell us what your research is focusing on?
My lab is interested in brain circuits and behavior in health and disease. More recently, we have become increasingly interested in neural mechanisms underlying emotion and cognition. We mainly focus on two key regions in the limbic system, that is the hippocampus and amygdala, to establish causal relationships between brain circuits and behavior. We combine the optogenetic or chemogenetic approach with different readout methodologies, including electrophysiology and cancer imaging in freely moving animals, to uncover the input-output organization of the network. We employ optogenetics assisted circuit making in brain slices maintained in vitro or in intact brain in vivo. Over the past decade, we have highlighted the relevance of GABAergic circuitry in both network and cognitive functions.
Dr. Lien, based on your expertise, what do you think are the important unsolved questions in neuroscience that might be solvable in the next 5 to 10 years?
There are several fundamental questions in brain science. I can foresee that one of them could be answered is the identification of the brain cell types. Today and in the near future, many new technologies will allow us to define the morphologies, gene expression, and function of single neurons at a high resolution and relevant to their function at circuit and behavioral levels. For instance, when I was a graduate student 20 years ago, we studied correlating single-cell morphologies with their gene expressions and electrophysiological features. At that time, we can only analyze 2 to 3 genes from a single neuron. Today, using the PAGE-seq technique, scientists can analyze thousands of genes in a quantitative way. With regard to the morphologic analysis, we fill the cells with biocytin during recording and then reconstruct their morphologies in slices or in juxtacellular recordings in the intact brain. Today, using a combination of mouse genetics, optogenetics assisted circuit mapping, and the tissue clearance technique, we can redefine the cell types based on their input-output logics more precisely compared to what we did before. Furthermore, the unbiased cluster analysis of all features also provide a very powerful tool in cell type classification.
How do you think computational neuroscience can aid experimental research?
Okay, I think this is a very good question. My short answer is yes. Computational neuroscience definitely can aid experimental research. When I was a graduate student, I was amazed by the Hodgkin-Huxley model for action potential. I think this is the best example for this question. Again, I want to use my PhD project as an example. Simply speaking, when I finished my first paper about functional characterization of voltage-gated potassium channels in fast-spiking interneurons in 2000, at that time, we almost believed that Kv3 potassium channel is required for fast-spiking action potential generation. However, we don't really understand what exactly the gating mechanisms of Kv3 under fast-spiking phenotype. To solve this question, I and my PhD supervisor tried to employ the computational approach to model Kv3 channels based on voltage clamp data acquired from my own experiments. Next, we combine the modeling with the dynamic clamp technique to inject the artificial Kv3 conductance into the fast-spiking cells, which was recorded in the presence of broad spectrum of potassium channel blockers. In this experimental condition, we can rescue the fast spiking phenotype by artificially injecting Kv3 conductance into the recorded neurons. The computational approach is very versatile and allows us to systematically alter the channel gating, such as the activation gating alpha, deactivation gating beta, and the inactivation gating. This approach led to the conclusion that fast deactivation, lack of inactivation, and high activation ratio of Kv3 channels are essential for fast spiking phenotype. Compared to the year 1951 or even 2000, our question today is beyond single action potential. Neuroscientists are facing more complicated questions, such as the network activities. Today, we can measure neuronal activities from hundreds of neurons in a small network. Using a computational approach, we can build up realistic network model, which is very close to the real network based on experimental data. So we can imagine if the model can simulate the neuronal activity, it may answer some key questions that experimental scientists cannot answer due to the limit of technologies. Furthermore, the model may provide some important clues or predict some important experiment for experimental scientists. So that's my answer.